mesodermal and ectodermal, endodermal tissues. From the ectodermal, I said earlier, so you get neural cells and epithelial cells. Okay. We will just transfer na kasi medyo blurred dito sa hindi ginagamit. So just for a while. This at its peak level. About 60 billion stem cells circulating. But they circulate as baby cells. They are not yet mature. When they mature, that's what we call they differentiate. So they can differentiate, okay? Like the bone marrow has two kinds of stem cells. The hematopoietic stem cells, which becomes red blood cell, white blood cell, platelets. And also bone marrow has what we call the mesenchymal stem cells. The bone marrow mesenchymal stem cells, that can, or bone marrow stromal cells, it can become cartilage, bone, muscles, connective tissue, and fats. Now, when on spatial, if they are being enhanced, spatial enhancement, they mature or differentiate, okay, into a different, they can become neurons. The bone marrow stromal cells, they can become neurons of the spinal cord and brain to repair brain damage or nerves tissues. That's why with the use of the stem cell system, I could see stroke patients, comatose patients, they improve rapidly, sometimes within hours. Okay, that's wonderful. Okay, now let's proceed. So that's what I use. The rapid cellular balancing of my system is actually stem cell enhancing system. And that is what I use in balancing the patient. Now let's proceed. From the embryonic stem cells, you get the three germ layers, ectoderm, which become the neural cells, gives rise to neurons, the brain, the spinal cord, and the glia. And epithelial stem cells, which becomes the skin and the hair. And then the mesoderm layer, you have the hematopoietic stem cells. This becomes blood cells, red blood cells, white blood cells, platelets. And muscle stem cells becomes muscle. And then other mesenchymal stem cells, osteoblasts, becomes osteoblasts and myocytes. Okay. And the endoderm, the other layer, gives rise to liver stem cells, and ultimately to liver, intestinal stem cells to become the intestine, gastric stem cells to become the stomach, and the lung stem cells ultimately to become lung. So they are organized that way, but that is what stem cells, when they mature, differentiate, they become what is programmed for them to become. But in spatial circumstances, trans-differentiation, they can become something else that is not programmed for them to become. Example, a bone marrow stromal cells is not programmed to become brain or spinal cord, but they can trans-differentiate with the proper nutrients, with the use of RCB, rapid cellular balancing, the one that I use. Okay. So here we have the stem cells. Okay. Over here is the common, these are their common origins. Okay. On the left is the lymphoid series, common lymphoid progenitor cells. This converts into T cells and B cells. The B cells, they produce antibodies, gamma globulins, okay, and so on. And the T cells, they're involved in phagocytic lysing or killing or devouring cancer and bacteria and so on. Then year over year, common myeloid series over here, this can become the granulocyte, monocyte progenitor cells, which ultimately become monocyte progenitor and to become monocyte. The monocyte ultimately can become macrophages. These are large white blood cells that can devour cancer. devour all the big cells. And over here, the granulocyte, monocyte progenitor cell can become granulocyte progenitor cells and ultimately to become neutrophil. This is according to their staining. When you say neutrophil or this granulocyte neutrophil, they are neutral staining. It's not acid, it's not basic. So light color, but plenty of granules, so that's called granulocyte. And it becomes basophil if it is base staining, Kool-Aid blue, or it becomes eosinophil if it is red or pink staining, Kool-Aid red, and mast cells. The neutrophils has also the work of phagocytosis or killing, eating up foreign bodies and bacteria, virus, and cancer. And the basophil and then the eosinophil increases when you have parasites. They tackle parasites, and the mast cells, they increase during allergies manifestation, okay? And they can cause you signs of allergies, urticaria, and so on. And then the common myeloid series progenitor will gives rise to erythroid progenitor cells, which ultimately become erythrocytes or red blood cells. And the job of red blood cells to carry oxygen all over the body, and it ultimately, the other one becomes monocyte, megakaryocyte progenitor cell, which ultimately becomes platelets. And the job of the platelets is to prevent bleeding. If you have dengue, your platelet falls and you bleed and bleed. If you have cancer and you are treated with chemotherapy, this will be totally arrested. All of these are arrested by chemotherapy, so you will bleed and bleed. Your red blood cell falls, you become anemic. So neutrophils, everything falls, and monocyte falls, T cells and B cells falls, and you cannot fight infection. So cancer patients, they die of what? Of infection and bleeding. And that can be reversed by the system that I'm using. I can stop the bleeding, I can regenerate the cells of cancer patients through stem cell enhancing. Actually, the ovary is the same. It's the same thing. Stem cells to megakaryocyte, erythrocyte, neutrophil, monocytes, B cells, and T cells. Tissue derivative of the three embryonic layers. Well, it says there, recall if I'm talking to the doctors or nurses, okay? So, sana in the future, ipag-pray po natin na ibitahin niyo ako ng PMA. Kung kayo yung nasa PMA, why don't you invite me? Kasi marami naman akong may tuturo na talagang to change our perspective sa health. Wala naman akong hindi naman sumasama ko. Pwede bang sumama ang loob mo sa magulang mo? Magulang mo yan. Sabi ni Lord, honor your father and your mother. Okay, I still salute PMA. Tissue derivatives are the three embryonic layers. Two main embryonic sources, okay? We have here the embryonic stem cell from the blastocyst, okay? And embryonic germ cells lines from the inner cell mass, which is the embryoblast cells from the another reach of the embryo after gastrulation, okay? Is shown here from the zygote, okay? You have there the blastocyst, the inner cell mass is the embryonic stem cells, and ultimately you are now forming the three germ layers, and then you get another one, the embryonic germ cell lines, and you still get the same thing, almost the same thing that I discussed. Okay, over here, all of this are actually what has been discussed already in the previous slides. For here you get the skin, the hair, the mammary glands, the lens, the lateral branch, spinal cord, epidermal, from neural stem cells, from neural, and the peripheral nervous system, the nerves. These cells were shown to be pluripotent, that is, capable of forming all mature cell phenotypes derived from the three embryonic layers, endoderm, ectoderm, and mesoderm. That is pluripotent, okay? Especially it's coming from embryonic germ cells, and even the embryonic stem cells from the blastocyst. The hierarchy of stem cell potency. If it is a zygote, day one, when the sperm unites with to form a cell, and that is surrounded by xenopotential, that is zygote, that is totipotential, 100%. All cells of the human body can be differentiated by, if you get it from the zygote. If it is from the embryonic stem cell, okay, which you can get from the inner cell mass of the blastocyst on the 5th day, post-fertilization, it is what we call pluripotent. It could become, the cell can become anything, endoderm, ectoderm, mesoderm, so almost everything in the human body. If it is adult stem cell, it is multipotent. Well, you get the adult stem cells from the umbilical cord of the baby, okay? Or you get that from the bone marrow, okay? You call it adult stem cells. Multipotent. Yeah, it's capable of being converted, but there is a limitation. For example, the bone marrow is limited to become mesenchymal cells and blood cells only. Okay, it's not designed to become brain, except when it is enhanced by transdifferentiation. It is only programmed to differentiate, but brain is not included. Now, once the stem cells are enhanced, okay, I was telling you earlier, at the age of 2, there are about 16 million stem cells circulating, and all of these are very, very active. And the highest production of stem cells is at the age of 2. Okay? And all of these can replicate continuously. Okay? They can rest, they can replicate, rest and replicate. And when I say replicate, they continue to divide to become baby cells. By the babies, they cannot work in the farm, right? They do not do anything. So once the baby mature, differentiate, it can become cardiac cell, it can become brain cell, it can become liver cells, and they are already functional. Okay. Now, adult stem cells are multipotent, but with certain limitations. But if it is already a progenitor cell, erythroid progenitor cell can become red blood cell only. Megakaryocyte progenitor cell can become platelets only. Monocyte progenitor cell can become monocyte only. It's limited to 1. So it is unipotent. Now when the cell is differentiated, no further differentiation potential. Okay? But as stem cells, they can renew. They can renew, self-renewing, okay? And as progenitor cells. It is the same. Stem cells potency by source. Totipotential cells, zygote. Okay, source, zygote. Pluripotential cells, embryonic stem cells, source, blastocyst. Multipotential stem cells, okay? Source, multipotential cells from embryo or adult brain, okay? Or also from the bone marrow and also the what we call and then limited differentiation potential, neuronal precursor, brain or spinal cord. Limited division potential, the brain fixating, neuronal precursor, sub-areas of the brain. Okay. And then the non-functional, non-mitotic neurons and differentiated neurons, specific areas of the brain. Okay. Fertilized egg. When it is fertilized, okay, it converts into totipotential egg. Okay, that is the zygote, and then ultimately to blastocyst, to pluripotent cells. Ultimately, these pluripotent cells, the inner cell mass, the inner cell mass in the blastocyst, can become neural cells, can become cardiac cells. That is the ectoderm, can become cardiac cells, the endoderm, and can become blood cells, the mesoderm. Okay. Endoderm cells are multipotent. Okay. The ectoderm stellar layers, okay, gives rise to the neurons of brains, the pigment cells, and the skin cells of the epidermis. Okay. The germ cells to the sperm and egg, and over here the zygote. 1 day, blastocyst, 5th day, and here what we show you the inner cell mass and the fate of the inner cell mass in the blastocyst. Okay. always the same thing. Mesoderm converts into cardiac muscle, differentiate into skeletal muscles, tubule cells of the kidney, red blood cells, and smooth muscles in that. Endoderm, internal layer to lung cells, alveolar cells, thyroid cells, pancreatic cells, and others. Totipotential cells can form the entire organism autonomously, 100%. Pluripotent cells can form almost all of the body's cell lineage, endoderm, mesoderm, and ectoderms, including germ cells. Let's say 90%. Multipotent cells can form multiple cell lineages, but cannot form all of the body's cell lineage. And that's about, let's say, 80%. Okay? Oligopotent cells can form more than one cell lineage, but are more restricted than multipotent cells. Maybe one or two. It can represent into one or two different cells. And unipotent cells or monopotent cells can form a single differentiated cell lineage. As I said earlier, bone marrow, stem cells, it becomes ultimately erythrocyte stem cells can become red blood cells only. Stem cells, what's their characteristics? They have a remarkable potential to develop into many different cell types in the body. So anything in the body, they can do. They serve as a sort of internal repair system, dividing essentially without limit to replenish other cells. So the job of the stem cells is like the singers, the carpenters. You know the singers, the carpenters, right? They are the carpenters of the body. Without stem cells, no repair. Okay. When a stem cell divides, each new cell has the potential either to remain a stem cell. So it can divide to remain a stem cell or become another type of cell with a more specialized function. So they can replicate, divide like plenty of babies, or they can differentiate, they mature, they can become liver cells to repair the liver, kidney cells to repair the kidney, pancreatic cells, and so on. Okay? So you'll see how things can be repaired even in cancer patients. They are distinguished from other cell types with two important characteristics. Stem cells are unspecialized cells. That means they have nothing to do in the body except just remain baby, capable of renewing themselves through cell division, sometimes after long periods of inactivity, even after 5 to 10 years. Okay. Under certain physiologic, stem cell enhancing, or experimental conditions, they can be induced to become tissue or organ-specific cells with specific functions. So when you have enhanced them, they mature, they go to the brain and repair brain damage. They go to the heart, repair heart damage. So that's stem cell, okay? All stem cells, regardless of their source, have three general properties. Self-renewal. It can renew. Capable of dividing and renewing themselves for long periods. Okay? Stem cells and special stem cells. They can keep on dividing. Okay? Unspecialized, they have nothing to do. Just like a baby gives rise to specialized cells. Okay, here's a master cell. That is stem cell. Unlike muscle cells, blood cells or nerve cells, which do not normally replicate themselves, okay, to replicate is to divide and form new stem cells with the same genes, with same DNA. Because blood is still the same, okay? Many times, para silang mga kambal. Okay? Kambal na nagpaparami, puro kambal lang, okay? Or proliferate. If the resulting cells continue to be unspecialized, like the parent stem cells, the cells are said to be capable of long-term self-renewal. Even up to many years, they can remain normal, if not excited. But if they're activated, especially